Well, welcome to our lecture, Human Genetics for Transition Year. So my name is Jacqueline Turner. I'm a genetic counsellor and I work with individuals and families who have genetic disorders. So I would meet with them to explain the genetic condition, how the condition is inherited, explain the results of their genetic test, and highlight in the family who's at risk of developing the condition and organise testing for family members who want to know if they carry the gene or if they're likely to develop the condition or not in the future. So I'm also chairperson of the Irish Society of Human Genetics, and in Britain they have a similar society called the British Society of Genomic Medicine, and it's a group of academics, researchers and clinicians who are all working in human genetics in Ireland. So what allows us to live? Inside our cells is this important molecule called DNA. And DNA is this double helix structure. So in 1953, Watson and Crick discovered that there was a double helix molecule present in every cell of our body. And it was through this molecule that traits were passed down from generation to generation. Up to then, we weren't sure how it happened, but we did know that traits were passed down in predictable ways, but we weren't sure how. The DNA contains our genes, and genes are recipes for proteins which direct all the processes in our bodies and allow our bodies to work and function normally. DNA obviously also allows us to reproduce, passing our essential genetics on to the next generation. So our body is made up of trillions of cells, and inside each of these cells is the nucleus, and the nucleus contains the DNA. And the DNA is a very long molecule, and in order to keep the DNA organized and not become a tangled mess, it gets wrapped around proteins, and that wrapped protein structure is called a chromosome. And we have 23 pairs of chromosomes, so the DNA molecule is split between those 23 pairs. So you have a piece of it on chromosome 1, a piece of it on chromosome 2, all the way down to our 23rd pair, which is our X and our Y. But if we actually stretch the DNA present on each of the chromosomes, we'd actually make up 6 foot of a molecule. And you can see how it would be very important to organize that molecule very well, because it has to squeeze into the tiny space of the nucleus of a cell, and it also has to be useful, so it has to be able to open up at certain areas so that the genes can be read and the proteins can be made. Our body is made up of different types of cells, but the DNA within each of the cells is the same, and that DNA originates from the very first cell, our fertilized egg. So when the egg was fertilized with the sperm, that very first cell then becomes a collection of cells, and each of those cells look the same, but they all have the capacity to become any of the different types of cells in the body. So the name for these cells, these original cells, is called stem cells. And you may have heard of that word, stem cells, because it's often discussed in the media. So the stem cells have the capacity to become any different type of cell. And now it's possible in the lab to take a sample, maybe a skin sample, and to reconvert it into a stem cell, to change it back into a stem cell so that it has the capacity to become a different types of cell. And how this is important in the future will be that, hopefully in the future we'll be able to make whole organs. We'll be able to make this stem cells become a liver cell, but then not only become just one cell, but to have the capacity to make a full liver. So the big problem at the moment is when we need an organ, if we have organ failure and we need an organ, maybe a heart or a lung or a liver, when we get an organ, we're getting someone else's organ, and it's very possible that our bodies will reject that organ as foreign. So people who get organ transplants often need very heavy drugs to try and prevent their bodies from rejecting the organ. But if we make the organ which is actually our own, so a copy of our own organ, well, then we're not going to reject it. So hopefully in the future, there will be the capacity, we'll have the ability to make whole organs that we can then use when we need them. And lately in the media, they've also mentioned that it's possible with these stem cells to maybe try and repair the cells that are broken in this spine. So if you've got spinal injury and if there's a problem with the spine, that you can actually use the stem cells to try and repair that. So this will have huge implications in the future. Our DNA is not organized in one long molecule. It's organized into separate molecules, separate little pieces, and they are present on our chromosomes, as I said. So we've got 23 pairs of chromosomes. So humans have 23 pairs, and they're numbered from the basis of their size, from number 1 down to number 22. And number 1 is the biggest chromosome, and number 22 then is the smallest one. And then you have an X and a Y, and girls have 2 X's and a boy has an X and a Y. So this picture we call here is called a karyotype. And here you can see that this person has the right number and amount of chromosomes. They've got 2 of each chromosome, and they've got an X and a Y, so they're a boy. And the chromosomes are banded, and the banding pattern is just because there's a dye that you can put on the chromosomes in the lab, and the banding pattern allows people in the lab to determine one chromosome from another. So you see chromosome 1 and 2, they're very similar sizes, and you could easily get them mixed up. But if they're banded, then they can very quickly, a cytogeneticist can very quickly determine one chromosome from another. And they can see then if there are little pieces missing, or if you've got extra pieces of chromosomes, because all of those things can actually interfere with our development. So it's very important to have the right amount of chromosomes, as I said. So here we have a little girl with Down syndrome, and you're probably familiar with Down syndrome. You probably know people who've got Down syndrome. And people with Down syndrome have an extra chromosome. So they've got an extra chromosome, a whole extra one. And you would think that having too much of a chromosome wouldn't be a problem, but actually there's a very fine balance in our bodies. And it's really important that we have the right amount, not too much or not too little. And people with Down syndrome have an extra copy of a chromosome 21. So if you look down here, you can see that she's got all the right number of chromosomes except for 21. You can see there's a whole extra copy of chromosome 21. And people with Down syndrome, they can have problems with particular areas. First of all, they've got a particular appearance that you can recognize somebody easily if they've got Down syndrome. They then can have problems with their heart, with their immune system, and they are at higher risk of developing a cancer called leukemia. And they can also have problems with their development, and they can have learning difficulties. It's important to say that no 2 people with Down syndrome have exactly the same problems. Some people can have more problems than others, and some people can have maybe significant heart problems, and other people have no problems with their heart. So it's very difficult to tell how that extra copy of chromosome 21 is going to how it's going to affect a person. But all we can say is that there's likely to be a pattern of particular medical issues that person could have. So here we have some pictures of a genetic condition called achondroplasia, and achondroplasia is a form of dwarfism. And on the left, you can see the actor Peter Dinklage, and you might know him as the actor in the Game of Thrones, or he's also the angry elf in Elf. And he has this condition, achondroplasia. And on the right, you can see that there are a set of non-identical twins, and one of the twins was born with achondroplasia. So you can see very clearly the difference between the 2 twins. So one of the twins with achondroplasia has shortened limbs. His torso, his body from his neck to the top of his legs is the same size as his twin, but his arms and his legs are much shorter. And you can also notice that there's a difference in the shape of his head and the size of his head. He seems to have a bigger head and a more triangular shape to his head. And they have a particular facial appearance as well. People with achondroplasia have normal intellect, and they have achondroplasia because they have a genetic change in one gene, and that gene is called FGFR3, a fibroblast growth factor receptor, which is important in making bone. And when you have a mistake in one of these genes, even though you have two, one from your mum and one from your dad, if you have a mistake in one of these genes, then you will develop achondroplasia. And the other thing to say is that people with achondroplasia, about 80% of people with achondroplasia have normal height parents. So that the change that's occurred on this gene, on this FGFR3 gene, the change has occurred for the first time in the pregnancy that gave rise to them. So although they had achondroplasia, they didn't inherit it from their parents. It was a new change that they developed when they were being formed. So here is another picture of another dominant condition called Marfan syndrome. And Marfan syndrome is a connective tissue disorder. Connective tissue is the fiber that supports and anchors and protects the body's organs and holds them in place. And when somebody has Marfan syndrome, there's a protein that's part of the connective tissue that isn't working as it should. And as a result, the connective tissue isn't as strong as it should do. And the body parts that are affected with Marfan syndrome are the skeleton, the eyes, and the heart mainly. And the skeleton, for somebody with Marfan syndrome, they have a particular appearance. They seem to be quite tall, and they have lax joints. They have, they can be double jointed or have lax joints. And one of the tests that we do is that their arm span is actually longer than their height. And there's another test that we do when you put your hand around your wrist, that your little finger and your thumb meet. So there are different tests that we can do to test the skeleton to see if there's skeletal features of Marfan syndrome. The other features are the eyes. People with Marfan syndrome, because of this lax connective tissue, their lens can fall out of place. And they can also get a retinal detachment, which is very serious and can lead to blindness. And the other issue, and the main issue for people with Marfan syndrome, is to keep an eye on their heart. And there is one very, very important vessel in the heart called the aorta, and the aorta is responsible for pumping oxygenated blood around the body. And so therefore it needs to be very strong. So when the connective tissue is weakened, the aorta can actually dilate, and sometimes it can actually come away from the heart, which is life-threatening. So for somebody with Marfan syndrome, you really need to keep an eye on their aorta and check that it isn't dilating and that it's as strong as it should be. And you'll see on the right a picture of the Olympic swimmer Michael Phelps. There is some question as to whether he has Marfan syndrome. He certainly has some of the skeletal features of Marfan syndrome, but he's actually denied that he has the condition. So if you look in your biology book might mention some more familiar dominant traits. And some of them are the ability to roll our tongue is a dominant trait over not being able to roll it. The other is a free earlobe. So you can see that your earlobe is completely free. And in this picture, you can see that somebody has a fixed earlobe. So the free earlobe is dominant over the fixed. And brown eyes tend to be dominant over blue eyes. We actually know that these traits aren't single gene traits, although they have mentioned it on our biology books. They probably are coded by very few genes, but certainly not just one gene. But still, it gives you an idea as to dominant traits, familiar dominant traits that we might have. So this is a picture of a baby who's got albinism, and he is missing a skin pigment called melanin. And this is also a genetic condition, but it's inherited a different way. It's a recessive condition. And therefore, this baby has no melanin. So both his genes, he has a change on each of his genes. So he's unable to make melanin. And there are different forms of albinism, but basically people with albinism are missing skin pigment, and they're missing the pigment in their hair and in their eyes. And they do need sun protection because they could burn very easily, because the melanin actually prevents our bodies from burning and protects our eyes as well. So before this little boy goes out, he would need sun factor, cover his skin, and he'd also need to be wearing some dark glasses. The second picture is of a newspaper article announcing the birth of a white baby to African parents. And the explanation is that the baby has albinism. And albinism is very common in certain parts of Africa. And in some places, probably as high as 1 in 1,000 babies are born with albinism. And the chance of a child being born with albinism elsewhere is probably about 1 in 17,000. And there's lots of superstition regarding albinism in Africa, and children can be persecuted because certain body parts are thought to have magical properties, or they could be ostracized because they're thought to bring bad luck. So one of my patients had told me that their child would be kept indoors and would not be allowed to play with other children and would not go to school and be educated like everyone else had they been living in Africa. So here we have a picture of twins, and these are identical twins. So they're formed when the fertilized egg splits in two, and therefore they have essentially the same genetic material. So here you can see that identical twins are formed when an already fertilized egg splits in two. But identical twins, actually, there are some differences in the DNA. There are differences in the non-coding, so the non-gene areas of the DNA. And also, identical twins have different fingerprints, and that's because the fingerprints are formed when the babies are in the womb. And it's the action of the fluid from around the babies that forms the fingerprints. It is a random event, and that means that any pregnant mother could be expecting a set of twins. It's something that can happen to any mother. Non-identical twinning occurs when two eggs are fertilized by two separate sperm. So basically, the DNA between non-identical twins is the same as for a brother and a sister, the same as for a sibling. And non-identical twins obviously are when you can have a boy and a girl twins. They'd be non-identical twins. And non-identical twinning there is a genetic component to non-identical twinning. And usually if you look on the mother's side, if there are non-identical twins on the mother's side or non-identical twins within the family, you know that there's a possibility. You probably have an increased chance of having non-identical twins. So we're just going to go back once again to the chromosomes, and the chromosomes, as I said, contain the DNA. But if we look at the DNA is actually made up of coding DNA and non-coding DNA. And it's the coding DNA that makes the proteins which our bodies need to work and function normally. And all proteins are made up of amino acids. So the amino acids make the protein. And so when amino acids are coded by a triplet repeat, so when the DNA opens up, there is a triplet code to start the sequence, and there's a code to stop the sequence. And then each triplet code codes for an amino acid. So in that way, the DNA sequence makes the protein. And our DNA sequence, there's only 4 bases that make up our DNA sequence. We have an A, T, C, and G, and those 4 bases and combinations of those bases make up our DNA. So the DNA present in each of our cells, a single set of this DNA present, is called our genome. And our genome is the difference, the reading of those bases, A, C, G, and T, all the way along from the beginning of our code, from the code that's from the DNA that's present on chromosome 1, all the way down to our X and our Y chromosomes. So it's the pattern of these codes that makes the genes. And in between the genes are non-coding regions. And before, we used to think that these in-between regions had no function, but now we know that they seem to have very important regulatory and other functions. And we're learning more and more about the areas in between the genes now. And that's a lot of our work is now trying to find out and discover what those in-between areas do. So if we write out our DNA code, we have there's 3 billion DNA base pairs which make up our DNA code. And within those 3 billion base pairs are 21,000 protein coded genes, so important genes. But if we look at that, there's only about 1% of our DNA is made up of genes. So there's a huge amount of our DNA that is non-coding. 99% of our DNA is non-coding. And I think this was a big surprise when we first sequenced the human genome in 2003, that we thought that we were much more complicated than we actually are. 21,000 genes doesn't seem a huge amount for the complicated organisms that we are. We also discovered that we are 99.9% identical to each other, so that although we look quite different to each other, we're actually quite similar. And that 0.1% difference is about 3 to 5 million changes. So there are millions of changes between us. So when we write out this DNA code, our genome, it takes a lot to write out 3 billion base pairs. And it actually makes up, if you look at a telephone directory and all the pages in the telephone directory, you mightn't even know a telephone directory, but we used to have directories of numbers, and it would make up about 200 different telephone directories. So the Dublin telephone directory by 200 would be one person's genome. So the big issue with genome and sequencing the genome is storage. How do we store all of that information on one person? And then when you're scaling it up to lots of different people, where do you store all of that information? So when we sequenced our genome, we then started sequencing other genomes and other organisms, and we discovered that we are actually very identical to other quite unlikely things. So we're 96% identical to the genome of a chimpanzee, but we're also 90% identical to the genome of a mouse, and we're 60% identical to the genome of a banana. So quite surprisingly, a lot of our genome is similar to other organisms which we wouldn't expect it to be. And how? How does this happen? So then when we thought we must have much more genes than other organisms, because we're far too much more complicated than that. So we must have more genes. And when we looked, we actually don't. So the rice that you eat has 38,000 genes, and a water flea has 31,000 genes. So our 21,000 is not even comparing with those organisms. So what's happening in us which is different to other organisms? How are we so complex? And the answer to that is really not in the number of genes, but in the way that we can process different proteins from the same gene. So it's now thought that we share such high percentage of our genome with other living organisms because we all arose from a common ancestor billions of years ago. And there are branches of genetics that look at evolution and different genomes from different places and dating time through the genome. And there are branches in Ireland that are actually looking at that, evolutionary genetics. So genome screening. So what is genome screening? So genome screening is taking a readout from you of all the bases that are present in your DNA. So writing out those 3 billion letters that make up what you are. And we can do genome screening by just a simple sample from you, either a saliva or a blood sample, and then we can read your entire genome, your coding and your non-coding areas. And genome screening is fast becoming a tool to enable better health management. And it is going to be the way forward, that our health is going to be organised based on our genome. So what are the benefits to genome screening? Well, first of all, you can determine the particular gene that's causing your condition. So before in genetics, we used to look at the person and try and decide what condition they had based on their appearance or the particular medical issues that they had. And then we would try and work out what condition they had and then look at that particular gene to see if we could find an alteration in it. Now, what we do with genome screening is that we can do panels of tests. So we can test, so I work in eye genetics, and we can test for any problems with the retina. We can test over 300 genes which can cause issues with your retina, with your eyes, and which can cause you to develop a blindness. So we can just take one sample from you and screen it for all the genes that are involved in retinal problems. We can also do gene screening to identify new genes. So we can look for different genes. So say you aren't on any of those panels and we didn't find the reason for why you're having problems with your vision. Well, then we can look at other genes that are outside the panels to see if we can identify a gene which can cause which might be the cause of your condition. So it might be a very rare gene that could be causing your condition. When we're genome screening, we can identify genes that could cause you problems in the future, or genes which we could screen you for. So say maybe if there's a cardiovascular condition, or if there is a cancer gene, we could then screen you for the types of cancers that can be caused by having that particular gene change. So we could actually identify genes that could cause you problems in the future. We could use it to treat genetic disease. So we now are trying to get we're trying to get the government to fund a particular genetic therapy for a very serious eye condition called Leber's congenital amaurosis. And we can now put the gene back into the eyes, into the retina of the eyes of those children who've got Leber's congenital amaurosis, and get this gene that they're missing working again, and increasing the vision of the people who have this condition. So we're actually putting working copies of the genes back into the eyes or back into the body to try and cure genetic eye disease or cure genetic disease. Something which I never thought we'd be able to do when I started out in genetics. We could determine factors that can prevent disease in the future, as I mentioned. There's also another branch of genetics called pharmacogenetics, and we know that there are certain genes which help us metabolize medicines, and that for some medicines are metabolized much quicker in some people than others. And if you need this medicine to maybe prevent you from developing or getting a recurrence of, say, a breast cancer, and you metabolize the medicine much quicker, well, then you might need more of that medicine for it to have the same effect. So it's really important then that you check that somebody is metabolizing the medicine at the right pace, and that you're actually administering the right amount of medicine for that person. We can also determine whether this medicine could have an adverse effect for you. So you may not be able to break down this medicine. So it may actually do you more harm than good. So there's a whole branch of genetics called pharmacogenetics, which is looking at that. It's very interesting. You can also determine your ancestry or your parentage. You can decide whether we've been asked, and although we don't do it, but it's possible to determine whether your parents are your parents. So sometimes people would like to know, is their father actually their father? And there are tests that you can do that. But you can also determine your ancestry. So where are you from? Where does your genome originate? Which is a very interesting area of genetics. You can also determine how people migrated in the past using the genome. And large databases have been used to fight crime, and I won't go into it, but there's a very interesting, and you might want to read about it, how they managed to find the Golden Gate Killer in the States was through a huge database, a genome database, which they use for ancestry purposes, and they found that the person they had the DNA from the person who had committed the crime, but they didn't know where had no more leads than that, and they actually matched his genome with the genome of somebody on the ancestry database, and they managed to find the person through their relative. So it just shows you how important genome data is. And I suppose I put this slide in just to back up my last slide when telling you about the genetic eye disease, the retina. Here's the retina, normal retina, and here's a retina that has a condition called retinitis pigmentosa. So the little black spots are areas in your retina. You have your photoreceptors, which allow you to see. And you can see that these areas, this doesn't look like a very healthy retina. It's not a nice red color like the other one. And these areas are these little blackened areas are areas of atrophy, which show that there has been cell death in the periphery of this retina. And the retinitis pigmentosa, people can have problems with their side vision. They seem to see quite well in their central vision, but over time they end up with tunnel vision. But there's actually about 80 genes which can cause retinitis pigmentosa. So in the past, we would have only looked at one or two more common ones. But now with genome screening, we're able to screen 80 plus genes in a row. So what are the concerns about genome screening? Well, it's your unique code. So there may be predictions within your genome whether you're likely to develop a particular condition or not. The condition may not be treatable. It may have a serious effect on your quality of life. It might even be life limiting, and you'd need to consider whether you'd want to know all of this information. Or even if you didn't want to know it, whether you'd want others to know it without you knowing it. So, the people who have done the testing, would you like them to know this information about yourself? So also there's privacy concerns. Obviously, there's nothing more identifiable for you to you than your DNA. So, people have concerns about how private this information is and where it's being stored, and who has access to that information. And the reason being is that, if you were likely to develop a condition in the future, like a serious medical condition, you wouldn't want to be stigmatized. You wouldn't want people to say, oh, I don't want Jackie working for me because she might develop this condition in the future. So it'd be much better for me to get somebody else who won't develop that condition in the future. So you wouldn't want people to be stigmatized because of what's in our genes. So we do have to be very careful. I mean, genome screening is here to stay, but we just need to make sure that it's safe and that people feel safe giving this information. So finally, I just wanted to make just a quick comment on the COVID-19 vaccination. There are different vaccines, but essentially the vaccines produce an essential protein of the virus called the spike protein. And you can see the virus here with the little spikes. So basically, the vaccines introduce the recipe, the DNA recipe or the mRNA recipe for this protein into your body, and your body then makes a spike protein. So it won't have any harmful effects to your body. It makes the spike protein, and then you make antibodies against that spike protein. And then when you get, or if you got infected by COVID-19 in the future, your body would recognize straight away the spike protein and start to attack the virus. So that's how it works. So I just wanted to leave you with that. So I hope you've enjoyed my talk. I just wanted to say that we understand it's been a very difficult year for transition year this year. We do have a competition. I'm just going to show you on the next slide our competition. It's the closing date is the end of April, but if you would like to write an essay or make a short film about genomics, we'd be delighted to look at it. The European Society of Human Genetics is running the competition, and the Irish Society of Human Genetics are offering prizes of 500 and 350 euro for our first and second winners. So just to let you know, this competition is run every year. So for the next couple of years, we'll be letting schools know when it comes out. So you're able to start writing it, and you've got a whole year to write it for next year. Or if you're incredibly efficient, you could get it in by the end of April. And this is my final slide, just really to tell you the closing date for our competition is the 25th of April, and that we have a top prize of 500 euro, a second prize of 350 euro, and you've been sent, or all the schools were sent, a poster explaining what the competition is and how to enter into the competition. The first line is, I propose to sequence the genome of because. So just to give you a starter on your essay, we'll be running the essay competition every year. The European Society runs it every year, so we'll certainly let you know. So anyway, listen, take care, and thank you for listening.